Yeah, let me know if that's not the case, Florian.
It looks good. Looks very
All
good.
right.
Parting
Uh,
ways.
All right. So my lab has been interested for many years in the process of chromosome segregation. So what you see here is a human cell that we've imaged at a high resolution microscope, where you see the process of chromosome segregation, in a cell, normal human cell, where we label the chromosomes with fluorescent histones in magenta and the centromeres in green. And these centromeres are really sort of the business end of the chromosomes in mitosis, because they assemble the kinetochores, and these kinetochores are essential for connecting the chromosomes to spindle microtubules and thereby segregating them. And we've done a lot of work in trying to understand these kinetochores and the way they function, as well as some of more recent work on centromere structure that I will very briefly touch upon in this seminar. But besides this, we're also very interested in when this process goes awry, and that is, of course, very common in cancer. And here we've imaged a U2S cancer cell line that was established in the 60s, I think. But this is a very common cancer cell line that has been used in many studies, and what you very clearly see here, and this is very common in this cancer cell line, but also in many other cancer cell lines, is this feature of lagging chromosomes, which is the most commonly observed segregation error in cancer cells. You see here this one chromatid basically remaining in the middle of the pack when a chromosome segregates, and this is very typical of what is known as a meritelic attachment, where a single sister attaches with its single core kinetochore to microtubules that come from opposite sides. Now, this phenotype of increased frequency of chromosome segregation errors compared to normal cells is what we call chromosome instability. And in the field, there's many definitions of chromosome instability. There can be genomic definitions, but here I refer to chromosome instability mostly as the dynamic process of enhanced missegregations in cells. So, like I said, also chromosome instability is used a lot in sort of the genomics field to basically define it as more larger scale chromosomal copy number alterations. And this is indeed the consequence of chromosome instability. So when cells missegregate, the daughter cells end up with more or fewer copies of chromosomes, and this is known as the state of aneuploidy, sort of a deviation of the copy number state from the multiple of a haploid set. Now, aneuploidy is very common in human development and human disease. For example, about half of all early pregnancy losses in humans are caused by aneuploidy. And of course, we all know also that there's aneuploidies that are viable and that have sort of very substantial developmental problems. And here, for example, is a karyotype of trisomy 21, one of the more famous developmental aneuploidies. And here on the left-hand side is a human oocyte that we've imaged going through anaphase, where you see many of the sort of lagging chromosomes, misaligned chromosomes right here. That is actually very, very typical when we do human oocyte analyses. But what we'll talk mostly about today is cancer chromosomes. And this is a very standard, typical karyotype of a human colorectal cancer cell line, where you see sort of this chromosomal chaos, right? There's a lot of copy number alterations that you can see, for example, from trisomy chromosome 7, a monosomy of chromosome 3, but also some larger chunks of chromosomes that have translocated to other chromosomes. And this is very, very frequent. So in a study from the Berukim lab and the Taylor lab a few years ago, quantifying the genomic alterations in cancer, including mutations and chromosomal copy number alterations, the most frequent mutation that was seen, and this of course is all known, and we all know this, is p53 mutations, but the 20 next most common alterations are all arm level or whole chromosome copy number alterations. So these are all these sort of green bars right here. And for many of them, including the ones that have this asterisk, the sort of the oncogene or the tumor suppressors that are associated with these chromosomes are not known. So what drives their copy number changes in cancer is not known. And I think there's a lot of people also here, I think in the audience, are very interested in understanding what sort of causes this selection for these copy number alterations in cancer. Now, So far, what I've told you is that there's a strong association of chromosomal copy number alterations in cancer and of chromosomal instability, so the dynamics of the missegregation of chromosomes in cancer. But what is the evidence that is causally implicated in cancer formation? So there's, I think, three main arguments for this. The first one is that there's a strong association, right? So there's really clearly a strong enrichment for certain copy number alterations in cancer. So that suggests that there's a selection for these particular genomic alterations. The second important argument is that there's a human syndrome called mosaic variegated aneuploidy, which is a disease that is very rare. So about 100 to 120 patients so far have been identified with this disease. And it's very typically associated with growth retardation, microcephaly, and cancer. And the defining feature is mosaic trisomies in a large percentage of the patient's cells across their bodies. And we've done quite a bit of work on this in the past, trying to understand the mutations that cause this disease. It's autosomal recessive, and about 40% of the cases have known genetic causes, and almost all of the mutations that we've known so far occur in genes that are involved in chromosome segregation. And the patients that get most often cancer are the ones that have mutations in these red genes right here that are all involved in the spindle assembly checkpoint. But there's also mutations that we haven't published yet in KNL1 and ZWINT1. These are sort of core kinetochore proteins, but also, for example, the centrosome protein CEP57. And the third line of evidence for chromosomal instability being important for tumor formation comes from animal models. And there have been many very sort of different types of animal models being generated already, like starting from 20 years ago, that have shown different mutations to model chromosome instability and have shown all kinds of different effects from oncogenic to tumor suppressive. And I just want to give you one example that comes from our lab, which is a mouse model in which we've designed sort of an allelic series that we can induce that increases the degree of chromosome instability with every combination of point mutations. And this is a model that we call the SIMQ model for Cre-inducible MPS1 mutation knock-in. MPS1 is a mitotic regulator, one of the spindle checkpoint genes. It's a kinase. And what this model does is we basically, after Cre recombination, we can induce an exon with a particular mutation in the kinase domain. And this mutation either reduces or completely kills the kinase activity. And these mutations are called TA, so this is actually a phosphorylation site mutant, or KD for kinase dead, which is one of the catalytic triad mutants. And if we combine different alleles, we can get a gradient increase in the degree of sin. And when we use this mouse model, and we do this in an intestinal cancer model with APC mutations, with a heterozygous APC mutation, we can see here in this increased degree of sin that this is counting the number of colon lesions, which are actually quite rare in this intestinal model for colon cancer. This is mostly small intestinal tumors that these mice get. When we start giving these mice additional sin, at the lower level, nothing much happens. And once we get to moderate and high levels of sin, you see that all of a sudden the colon starts to be completely full of polyps. So this can be quite oncogenic, and we also showed in this paper that's already now published 6 years ago that by itself chromosome instability, so without this APC mutation, can also cause colon adenomas. So these I think, are the sort of the main arguments for thinking that chromosome instability, and then most likely also the related aneuploidy, can be oncogenic. So our lab has been, like I said, interested in various aspects of this. So over the years, we've done a lot of work on the molecular mechanisms of chromosome segregation. So this is really sort of the fundamental basic research on the proteins and the regulatory principles of chromosome segregation. And then we use those insights to also understand the origins of chromosomal instability. So why do cells increase missegregation rates? What is going wrong in those cells? And then 2 lines of research that deal with how does this contribute to cancer development, and also to embryonic developmental defects. All right, so there's many questions that the field studies, and there's many fantastic labs that work on various aspects of those different questions. And I just want to highlight 4 questions that I want to further go into in this seminar. The first 2 questions I will briefly touch upon, and the last 2 questions will be the bulk of the seminar. The first is, how do lagging chromosomes in cancer cells arise, right? I said that lagging chromosomes are the predominant let's say, form of missegregation in cancer cells, and we still don't really understand how they come about. Then there's this interesting concept of aneuploidy patterns. So different cancers have different copy number alterations, let's say signatures, and we want to understand how they come about. And then the second half of the seminar, I want to go into how do cells cope with chromosome instability and aneuploidy, and we're particularly focused on p53. Because it has been suggested to be sort of the most important mutation to allow cells to cope with chromosomal instability. And finally, I want to get back to this mouse model because we have some new insights into how chromosome instability can contribute to tumor formation. All right, so to start with the first 2 questions very briefly, because these are studies that have been published. We recently, 2 years ago, published a study that was basically about the centromere and the centromere structure. And what I mean by centromere structure is both based on sort of chromatin configuration sequencing, as well as super resolution microscopy. We found that the majority of centromeres in human cells, in mouse cells, in chicken cells, in various species that we looked at, is not a single folded chromatin structure in mitosis, but it is a doublet structure. And this doublet structure is stabilized by cohesin. But what is very interesting is that it also is not only a double structure, but it binds to 2 main microtubule fibers. Now, we don't really yet completely understand why this is the case, so we're actively studying this. But what we did see is that this would create a situation that explains merothelic attachment. If you have 2 fibers that need to connect to the same sister chromatid and to the double structure, you can also imagine how this could go wrong and create a merothelic attachment that would then sort of be this driver of lagging chromosomes. And one potential way to do this, of course, if you have structural alterations in the centromere itself, but also if you have any issues with cohesin, for example. And just to show you what this would look like, this is again the U2S cell line, and this whole study was done by a very talented postdoc lab, Carlos Sacristan. So this is an ISIM microscope image, so it's super resolved, sort of double the resolution of normal microscopes, because we needed to see sort of these 2 different domains in the centromere, which is here is a very, I think, beautiful example of a lagging chromosome, where you can see these split centromere domains, basically really suggesting that there's a merothelic attachment happening here, just like sort of depicted in this cartoon on the right-hand side. So one hypothesis that we have is that potentially the centromere structure, either by issues with condensin or other features that determine centromere structures, or the cohesins, is somehow defective in cancer cells to allow them some destabilization of the centromere structure that could cause merothelic attachments to be basically stabilized and therefore cause lagging chromosomes. And we're investigating this. Another sort of hypothesis that comes from this is that you could also imagine that if there's any breaks or break proneness, let's say in the centromere under these conditions, for example, by replication stress, that under these conditions, this could lead to arm-arm separation, and might be an explanation for the high prevalence of chromosome arm alterations in cancer as well. And this is something that we're also investigating. All right, so the other questions, second question I had was about cancer aneuploidy patterns. I won't go into this very deeply, but I just want to highlight one study that we published a number of years ago. And this has to do with the question of how do you get from a diploid cell, in the healthy tissue, to one that has a particular copy number alteration. And this is an example of colorectal cancer. where at the end stage, we see a particular chromosome alteration, such as trisomies of chromosome 7 and 8. But if you would look at breast cancer or kidney cancer, for example, those copy number alterations would be different. And there's some beautiful work by various labs that, of course, suggests that this is mostly driven by selection for whatever sort of gene networks is important for the stem cells of that tissue. I think we still don't understand completely sort of what drives this and what is the actual mechanism. But what I wanted to just highlight here is that there's also this idea of, sorry, I skipped it too fast. There's also this idea of the mutation rates that allow cells to undergo selection for particular copy number alteration. What I mean by this is that chromosomes don't segregate, sort of the chromosomes that missegregate are not always the same ones. And they're also not equally missegregating in every cell division, meaning that chromosome 1 or chromosome 8 do not have the same missegregation frequency. And the way we show this is by single cell sequencing, but also by live imaging. And what we found is that certain chromosomes have particular positions in the interface nucleus. This was known from many studies in the past. But what we found is that when cells enter mitosis, the location of those chromosomes in the nucleus actually dictates the probability with which they would missegregate, for example, in cancer cells, but also under experimentally controlled conditions. And this has to do partly with their sort of more difficulty in getting attachments, proper attachments established, but also bypassing, for example, the spindle pole and things like this. And one of the ways that we show this is by labeling using fluorescent Cas9 different regions of chromosomes. For example, the big dots here are chromosome 9, the smaller dots are chromosome 1. And then we could basically say what happens to the chromosome in anaphase, and how does this relate to the starting position of that chromosome. And when you quantify all of this, you can see that if you just take chromosome 1, which is most often peripherally located, that's its, let's say, the highest chance when you look at interface nuclei that chromosome 1 is peripheral, then we see a relatively high segregation error rate compared to when occasionally, in this case, 31 out of the 150 or so cases, when it starts centrally, then its segregation error rate is much lower, and vice versa for chromosome 9, that is most often centrally located. Its segregation error rate becomes much higher when occasionally it's peripherally located. And of course, we had some other pieces of evidence for this model. So what we think is that it's not unbiased where the which chromosomes missegregate, which ones become aneuploid in the daughter cells, and which ones also become entrapped in micronuclei. So larger chromosomes, the more peripheral chromosomes, much more frequently end up in micronuclei, for example. All right, so what we don't know is how important this is in getting to those aneuploidy patterns in cancer. I think probably the selection for the karyotype is a much more important driver, but it will influence the path with which the chromosomes end up getting there. And we're trying to model this, amongst others, in our collaboration with Nenad Paven and Iva Tolic in Croatia. All right, I want to talk now more elaborately about two studies that we put on bioRxiv in the last few months or so that deal with sort of aneuploidy tolerance or adaptation to aneuploidy and how chromosome instability can contribute to tumor formation. But let's start with the first one. So as I showed you earlier, 53 mutations are very common in cancer and are the most common in cancer. And then the next are all the chromosomal copy number alterations that are listed here in this graph. And there's been quite a few studies that have suggested that that these two are associated, so that 53 mutations are enriched preferentially in aneuploid cancers. And this is a study by Alison Taylor from a number of years ago, where she analyzed the mutated genes in cancers with different copy number alterations or in aneuploids or euploid cancers. And 53 was by far the best associated mutated gene in these aneuploid cancers. Now, we wanted to look into this a little bit more because there's been a bit of sort of confusion about exactly the role of p53 in tolerance to aneuploidy. The first, I think, is that the prevalence of aneuploidy is far greater than the number of the mutations in p53. So about 80 to 90% of cancers have copy number alterations, and about 40 to 50%, depending on how you measure it, of cancers have p53 mutations. So that doesn't really match. So there must be more to the story than just aneuploidy or not. And the second point is that there's quite inconsistent data from in vitro models. So depending on which cell lines you use, or animal models, or for example, organoids versus 2D cultures, whether or not you need p53 to become aneuploid and to survive aneuploidy is different. And so we wanted to dive a bit deeper into the cancer genome data to see if we could find could verify the association with p53, and if there's any features about the types of aneuploidy or certain features of aneuploidy that would explain the p53 association a bit better. So we teamed up with Teresa Davoli over in New York, and this was all done by a very talented student, Joanna, who just graduated. And what she did is she took from a data set published by Teresa of TCGA data tumor sets that were sort of screened and filtered for copy number alterations and p53 status, etc., whole genome duplication information. And then she basically made all kinds of subcategories where we could classify the tumors as euploid or aneuploid, and then whole genome duplicated or not, and then the p53 status. And we included not just p53 mutation status, but also p53 mutation phenocopy data. So this is transcriptome data that is transcriptomes that sort of are similar to p53 mutant transcriptomes, and are usually sort of caused by mutations in MDM2 or 4 or USP28 mutations. And this is about, I think, about 10% total or so of the tumors that were originally classified as p53 positive based on the mutation status, but end up being p53 negative based on the transcriptome status. And this sort of amounts to about 50% of the tumors then having a p53 deficient assignment. So this is a nice data set to sort of explore a bit deeper. Can we see any association of p53 status with particular features of aneuploidy? And the first thing that Joanna did was just look at aneuploidy, yes or no, and whether or not a tumor has a p53 mutation. And if you look at the pan cancer sample, then this is basically all the aneuploid cancer samples here on the right-hand side, and the euploid ones are on the left-hand side. p53 status is also in the next slides. Sort of the reddish color is p53 deficient, and the gray color is p53 proficient. And indeed, you can see p53 mutation enrichment in the aneuploid samples. It's about threefold higher in the aneuploid samples. But if you look at sort of cancer types, you can actually see quite a few cancer types where euploid and aneuploid either don't have very many p53 mutations, or, for example, like this one, where you have very high, at least equally high numbers of tumors in the euploid cancers that have a p53 mutation as in the aneuploid cancers. So yes, if you do the pan-cancer analysis, it's very clearly associated, but if you look at cancer types, it doesn't always hold up. But of course, it's a very simplistic analysis, right? It's basically aneuploid, yes or no, but there's many features of aneuploidy that we here just kind of ignore. So we wanted to dive a bit deeper into that. So there's different features that we looked at. First one, and that's something that I think would make a lot of sense, is that does the degree of aneuploidy matter? So what I mean by that is you can have simple aneuploidies, for example, one trisomy and the rest of the chromosomes are all diasomic, or you could have more complex where you would have a gain of a chromosome, but also losses of chromosomes, etc. So that would be the degree of aneuploidy. And then, of course, you would have whole chromosome copy number alterations versus arm level. And these arm level alterations, of course, require DNA breaks with which p53 might be more associated than whole chromosome alterations. And finally, we also looked at gains or losses. So it doesn't matter whether or not you gain chromosomes or lose chromosomes. And Everything I will show you from now on is on the tumors that have in which the whole genome duplication has been excluded to simplify the analysis and don't have this confounding factor of whole genome duplication. Okay, so the first way we looked at simple or more complex aneuploidy is by looking at the fraction of genome altered. And the reason for this is that we think that most likely the impact of chromosome instability on the cells has to do with the number of genes, or at least sort of the size of the chromosomes, the number of genes, the number of protein coding genes on these chromosomes, and the proxy for this could be the fraction of genome altered. Now, what this means is that we just basically looked at the number of nucleotides that have been altered in these cancers, with the reasoning that, for example, a loss or a gain of chromosome 1 would be equally impactful as a loss or gain of both 6 and 18, because they have the same amount of DNA in the end. And so when we look at this and we just classify the tumors on fraction of genome altered, we indeed see that p53 deficient cancers seem to have a higher FGA. And what we do here is that a loss or a gain is considered the same fraction of genome altered, right? It's basically just the change from the normal state. But again, if you then look at different cancer types, a slightly more nuanced picture comes out, where indeed there's quite a few of them that have a fraction of genome altered in the p53 negative that is higher than the ones in the p53 positive samples from the same cancer type, right? So that's plotted here on the y and the x-axis. But also quite a number of tumors that have equally high fraction of genome altered in the negative versus the positive p53 samples. And there's some that can reach very high levels of fraction of genome altered, so very high aneuploidy, and that is equally high in either the p53 negative or the p53 positive cancers. So also here, yes, pan cancer, it looks to be this trend of p53 mutation will allow for the selection of higher aneuploidy states, but there's very clear tissue specificity in this, which is, I think, kind of fascinating. Like, how can some of these cancers get such high aneuploidy without a p53 mutation? So we also looked into arm and whole chromosome copy number alterations, and I won't show you the data, but we didn't really see a big effect there either. So there, also pan cancer didn't seem to be any clear preference for whole or arm level copy number alterations in the p53 deficient versus the proficient states. But here's another example, a slightly different depiction of this situation of the copy number alterations, sort of the degree of copy number alterations. Here, what we did is we ranked the individual tumor samples now, and I just showed you two examples of bladder cancer and endometrial cancer. We ranked them based on fraction of genome altered here on the x-axis, and we color-coded them again according to p53 mutations. And you see, for example, here that the very highest aneuploid samples in bladder cancer are all p53 mutant. But you also see, for example, in the endometrial cancer, that this highest quartile of FGA does not seem to be much more enriched for p53 mutations than the third quartile in this case. And I think also some notable things that you see from plots like this is that you can have p53 mutant samples that are very lowly aneuploid. So that says that p53 mutations are not sufficient to drive aneuploidy appearance in these cancers. And of course, you will have very high aneuploid samples right here that are p53 proficient, basically making the point again of these tumors somehow deal with high aneuploidy without inactivating p53. So the final one that I just mentioned, the arms versus the whole chromosome, we didn't see a big difference there. But when we started looking at losses versus gains, we saw something sort of more generally interesting. So what we did here is we took all cancers that have gains, and they could have only gains or gains and losses. That doesn't matter to us, but we only looked at the fraction of the genome that was associated with chromosome gains. And we will plot this. We don't see any difference between p53 negative or p53 positive tumors. So the fraction of the genome that can be gained in either situation is similar. But when we looked at the fraction of the genome that is lost, so only the losses, then you very clearly see that p53 negative tumors are have a higher fraction of the genome that can be lost, as if p53 mutation sort of biases for the survival more of losses than it does for gains. And here, I think it's quite striking if you plot the individual tumor types. This would be sort of this line would basically mean that the average of the fraction of the genome that can be lost versus gained is the same in a particular cancer. You see that almost all cancers are shifted to the upper left side of this graph, meaning that p53 mutations are causing losses to survive much more than in the p53 positive samples. So we wanted to sort of explore this a little bit further. So what we did here is we did what we call a delta FGA. So we subtracted the fraction of the genome altered for losses from those of the gains, and meaning that if you're below the 0 line, you have lost more of your genome than you have gained on average. And then we again plotted all these fraction of genome altered along the x-axis right here. And you can actually see this ovarian cancer, in which we know that almost all cancers are p53 mutants, but you can also see that almost all of those cancers have predominantly more losses than they do gains. So the delta FGA is below 0, meaning they have more losses than gains. And this endometrial cancer, again, is also a beautiful example where you see that those tumors that have predominantly more losses than gains are very much enriched for p53 mutations. So that really suggests that p53 mutations preferentially biases for the selection of losses than anything else. So we wanted to also look into sort of more experimental settings, like can we recapitulate something like this when we cause chromosomal copy number alterations in p53 positive versus negative cells. And what we've done here is we did this experiment RPE1 cells, and we teamed up with Uri Ben David's lab, who did the same experiment in MCF10A cells. And what we did is we knocked out p53 or not. We gave them SYN by, in this case, MPS1 inhibitors. And then we let them recover for different numbers of weeks, and we did single cell DNA sequencing, a method that we call single cell karyotype sequencing, to basically look at copy number alterations. And this is an example of RP1 cells 4 weeks post chromosome instability. So you see all kinds of copy number alterations. So these would be, every column would be a chromosome from chromosome 1 to 22 and X, and every row is a cell. If it's green, it's disomic. If it's red or yellow, for example, it's copy number gains. If it's purple, it's copy number losses. You can already see that the p53 mutant samples have more losses than the p53 proficient samples. And if we quantify this for the different cell types, here on the right-hand side, you would have the gains that we can see in the RP1 cells or the MCF10A cells, p53 mutant versus wild type. You see that there's no real difference, or there's even a drop from the p53 deficient to the proficient, and the losses are clearly now more enriched. So every dot here is a single cell. So it really does suggest that p53 inactivation preferentially enriches for chromosome losses after chromosomal instability. So sort of to summarize this, what we showed, at least in these TCGA analysis and with a little bit of this experimental data, is that we think that p53 proficient cancers tend to have more simple karyotypes, but this is very clearly cancer type specific. And p53 deficient cancers seem to have slightly more complex aneuploidies, and also are preferentially associated with chromosome losses. And some of the things, of course, that we're very interested in is what happens with these p53 proficient cancers that have very high aneuploidy, such as these ones that are listed right here. So how do these cancers adapt to aneuploidy? And we don't think it's p53 pathway inactivation, because we also profile this p53 mutant phenocopy state. And so there is likely something else. Either these tissues do not readily activate p53 upon aneuploidy, or there's other mutations or alterations that allow them to cope with it. And for the losses, of course, what causes the preferential survival of cells with monosomies, chromosome losses in p53 deficient cancer? So there are some suggestions out in the literature, but I think we don't know for these particular states why 53 is so strongly associated with monosomies. All right, for the final 10 minutes or so, I want to go into the final bit, which is how does chromosome instability contribute to tumor formation? And I want to get back to this mouse model. So I showed you that we have this mouse model where we can induce sort of different degrees of chromosome instability in different tissues because it's a Krelux system. And what we really wanted to do is we wanted to see what do the different degrees of chromosome instability do, and can we have a model in which we can monitor this over time? And what we chose here is sort of a very classic skin tumor model. So this is the model in which we have a DMBA treatment that generally just induces a KRAS mutation. And this is followed by TPA application, which has all kinds of effects, but by and large, a very important factor in TPA treatment is inflammation, but also there's some regenerative responses and other responses that it induces. And this then creates skin tumors on the back of the mice, where the application of these two compounds was done. And then in our case, we give them chromosome instability at either, let's say, a mock situation or the different degrees of chromosome instability. And then we monitor over many, many weeks the tumor formation. And the big benefit of this is that we basically, because we can look at them and we can measure the size, we can measure the number, and we can do this over time, this is a very interesting model for us. Like I said, also this was recently put on bioRxiv, and this was all work by a previous postdoc in the lab, Nanette, and also a talented student that's still in the lab, Simona. So first, of course, is we had shown that this model does exactly what it is supposed to do in the intestine. Basically, the different mutations cause different degrees of sin. So we wanted to make sure that this is also the case in the skin. So what we did here is we made skin organoids from these mice, induced the mutations, and then quantified by live imaging of these organoids going through cell divisions, the degree of missegregations. And you can see here the different levels of chromosome instability from 0 to very, very high. And indeed, you can see the sort of this graded increase in chromosome instability levels, especially going from sort of the low levels to the moderate and high and very high degrees of chromosome instability, where you can see that the ones in which basically MPS I kinase activity is knocked out have almost uniformly very severe missegregations. All right, so then we monitored the tumors, and this is an example of where we quantified the average number of tumors here on the y-axis, and this is the time. And then we also classified them based on sort of categories of tumor sizes. And this is sort of the typical growth of the skin tumors if you don't give them chromosome instability. Now, if you give them a little bit more chromosome instability, from very low, and I just give you two examples here, very low and moderate, you can see that in very low chromosome instability, you do see that the number of tumors increases a bit, but the volume doesn't really change very much. And also the onset is the same. But if you go to higher levels of chromosome instability, now you see that the onset becomes much earlier, and also the size of the tumors becomes bigger. And when you do this for all the different chromosome instability levels, you get a plot like this. I'll take you through it. So on the x-axis is the time of tumor onset. On the y-axis is the number of tumors, and the size of the ball, let's say, is the tumor size on average for these different mice. And of course, this is a cartoon, so this is just sort of a representation of what the data looks like. So if you don't have any chromosome instability, your tumor onset is relatively late, the tumor size is sort of, relatively small, and the number of tumors are relatively small. And when you increase chromosome instability, what you first get is that the number of tumors goes up, but nothing else changes. Then you go to the moderate chromosome instability, and now basically a lot of things start happening. Tumor onset increases, so it's earlier. The number of tumors is still very high, and the tumor size starts to increase. And this seems to be sort of the optimal, let's say, bad state, because if you then further increase chromosome instability to high and very high, although you still get earlier tumor onset and the tumor size is still big, the number of tumors starts dropping. And I will get back later to what we think is happening here. So one thing that was actually surprising, sorry, I have to go back, is that what I told you is that the very high chromosome instability levels really are almost uniformly severe missegregations. And from all models that we know, this would cause cell death. And so why do these mice even develop tumors at all was kind of puzzling to us. So the first thing that we decided to do was go back to the tumors and make sure that the mutations that we had induced were in there. And so what we did is we RNA extracted RNA from the tumors, and we did RT-PCR and Sanger sequencing to assess these MPOS1 allele mutations that we induced. We did this after 1 week of induction. So we applied tamoxifen, we took out part of the skin, and we sequenced for these mutations, and we actually verified that they were very efficiently, just like in the organoids, they were very efficiently induced on the backs of these mice as well. So for example, in a TA-KD combination, we always found about 50% of the alleles being TA and 50% being KD. When we, however, looked at the tumors, we noticed that only the low SYN tumors had the right mutations that we induced, but the higher SYN ones did not. They always lost one of the two alleles. And this was very clear, for example, in the TA-KD situation, whenever we sequenced, we only found wild type TA or wild type KD. And so what that means is that the tumors that arose from the higher SYN levels, so these guys that have a very clear, distinct phenotype, right? Bigger, more tumors, but genotypically, they were the same as the low SYN tumors that we induced. So what is going on there? The one hypothesis that we had is that maybe very rarely the recombination is not efficient enough to induce both alleles. And so, for example, in this situation here, you would have one cell that is still low or very low SYN, and that somehow this cell is stimulated to grow out much faster and earlier because of the presence of these very high SYN cells. So these clearly disappear because we don't see them back when we do the mutational analysis. Yet they have a profound effect on the growth of these low SYN cells because in the end, they have bigger tumors that also start earlier. So we want to understand this a little bit better, whether this was the case or not. So we wanted to understand what is actually the high SYN doing in this situation. So the first thing we thought is, well, maybe, and this of course has been reported by various other labs, it induces inflammation, and because this high SYN causes inflammation, maybe the inflammation helps the lower SYN cells to grow out sort of with a vengeance. So what we did is we took out the TPA from the protocol and just induced chromosomal instability to see if it could substitute for TPA. And what is kind of striking is that here you have the tumor formation in the mice that only get DMBA, so no TPA, no chromosome instability. You see only very few tumors, very small tumors, and they arise really late. We give them a little bit of chromosome instability, nothing much happens. But then we give them this moderate chromosome instability, and now you very clearly see all kinds of tumors arising. And if we just apply moderate SYN, nothing happens to the skin very much. So it's really this combination of DMBA and moderate SYN that all of a sudden creates a lot of tumors. All right, so does this mean that instability is indeed an inflammatory response? So what we did is we then treated the mice with an inflammation inhibitor, so carprofen, which is a COX-2 inhibitor. We put it in drinking water. As a control, we did it in the sort of the classical model of DMBA, TPA, and then inhibiting inflammation. Or we did DMBA, chromosome instability, just like here, and then we inhibited inflammation. So in the control, indeed, we see that TPA does induce some inflammation because if we add carprofen to the drinking water of these mice, we see that the number of tumors and the size of the tumors generally just decreases. We also see that it's not complete, even though when we do all the immunohistochemistry, it looks like really there was a very efficient suppression of immune infiltration in these tumors. There is some other effect of TPA, I think, that is in play here, but there's a strong decrease by carprofen, so it works. If we do this, however, on the moderate chromosome instability that is applied to the DMBA situation, we don't really see much of a difference. So still early onset, still big tumors, still many tumors. So it looks like what chromosome instability is doing and what it's mimicking, let's say in the TPA situation, is not really inflammation, but it's probably something else. So what could that be? So we looked at a bit closer at the tissue, and we could see that there's clearly hyperplasia if we just give them chromosomal instability. So we see some things happening here in this messenger trichrome stain, a thicker epidermis, and we also saw some more sort of cell divisions in these early states. And then we did RNA sequencing on these backs. We did sort of different days, but I'll show you the day 7, so 1 week after we applied tamoxifen for 5 days. And we noticed, amongst others, that there was an increase, a clear increase in proliferation, but also a decrease in cytokines and some other immune signatures. And so we wanted to verify this using immunohistochemistry. So what we found indeed is that on the backs of these mice, we see a regenerative response. So this is a Hedgehog pathway, a transcription factor that in the moderate chromosome instability is very clearly sort of enriched in these backs. And you also see here this sort of much thicker epidermis in these skins. So there's a regenerative response. And also we see this immune exclusion. So here's a CD45 immunohistochemistry of the wild type skin, and this is this moderate SYN state, and here's a quantification of the different degrees of chromosome instability where we quantify the immune cell infiltrate. And basically what you see is that you have a decrease of CD45 cells in these chromosome unstable backs of these mice. So we think we have a combination of sort of a proliferative response that could be because of either cell death or some other thing that these high SYN cells are doing, that is really stimulating these low SYN cells to grow out and form a tumor. And so what we think is going on is that under normal conditions with low SYN, you would have that we induce by tamoxifen, you have these sort of slowly growing out because these are the cells that have the right sort of state for copy number alterations or micronuclei formation that is beneficial for these cells to proliferate or to compete. But this is a relatively slow process, and that they are actually benefited by very high chromosome instability cells surrounding them, in some way inducing their outgrowth. So there's more apoptosis that we saw in the skin. I didn't show you this. There's a regenerative response, suppression of inflammation, and this together creates this outgrowth of the lower SYN cells in the tumor. And in fact, what is kind of surprising to us was that we also saw this in other mouse models where we looked at the memory tissue, intestinal tissue, and mesentery tumors, where when we end up sequencing the MPOS1 mutation in the tumors, even though we induced high SYN and the phenotype is more severe than the lower SYN, the tumors consisted of low SYN cells. So with that, I want to thank several people. I think I already mentioned Joanna and Simona, who did the p53 and the mouse work, several other labs, collaborators like Teresa and Uri Ben-David, but also Hans Clevers, who's been very helpful with the skin organoid work, for example, and several others, and of course, all our core facilities and funders. And I want to thank you for listening to me for almost 50 minutes.
Yeah, thank you very much. That was excellent. Now, hopefully we have some questions. You can put your hand up, and Aurelie will be first. She had her hand up first. Or you can just write in the chat, and I will read out to everyone if that is okay for you. So have a think while Aurelie asks the first question.
Thanks a lot. Very beautiful work. Thanks a lot for accepting our invitation. I have a question on whether you normalize for the ploidy, so for the link with so between p53 inactivation and the losses, because you could imagine that so if you have genome doubling, which is linked with p53 deficiency, then the losses will be tolerated. So I was just wondering whether this was normalized.
Yeah, so we basically we take sort of this average of triploidy in these cancers. So this has all been done by Teresa. We don't really normalize for the ploidy, so we take them basically based on sort of this triploid state, and then we later just look at the different chromosome copy number alterations.
Stefano.
Hi, Gert. Very nice. I think I had a similar question. It was more about the start employee that you have in the p53 wild type versus the knockout. Because if you have already losses to start with, and then you use MPS1, you're basically reshuffling a population that is already skewed towards losses.
Oh, you mean in the experimental situation?
Yes.
Yeah, so no, basically we just sequence all the cells, and we do different analysis. So you can basically say we baseline it for disomy or more, but this is basically just a standard analysis where we have, in this case, we have disomy, or let's say diploidy as a standard, and then we look for sort of differences. But it's true that we might miss a few cells there because we do have to, let's say, fact sort them based on some ploidy width, right? So we don't want to go into S phase or G2 phase cells, right? Because then you have no idea what you're sequencing. So we try to do G1, and then we do sort of have this ploidy range. So we might miss sort of the more extreme cases. That is true. Yeah.
Thanks.
Thanks, Stefano. Teresa.
Hi, Geert. That
Hi,
was a great
Teresa.
great talk, especially because all the part on the inflammation, totally unexpected. I would not have expected that, right? So
No, me neither.
that's why it's super interesting. I was just wondering, is this decreased in CD45 infiltrate present only if it's in conjunction with TPA? So it's always the TPA minus sin and TPA plus sin that, or I think I missed, yeah.
Yeah, so in that case, we didn't do TPA, we just did DMBA and sin.
Okay, sorry, okay,
Yeah.
okay.
So we took TPA out because it does so many different things. We just wanted to know if we apply sin to these backs, what happens, right? So did we do it under the oncogenic condition of DMBA, let's say, and then we very, very clearly see immune depletion after a week, and this is still persistent after 4 weeks.
So again, this is totally different than what we expect based on the literature.
Yeah.
So what do you think is going on?
We don't have any data really to speak to what is going on really. So we try to dive a bit into the RNA sequencing to see if there's any obvious thing. We have done an experiment in the intestine where we looked at something similar. We did single cell sequencing there for after I think about 3 weeks or so of the induction. And there what we do see is what we think is a sort of a chronic interferon response. So there has been some literature that says that chronic interferon response is immune suppressive, and this might be caused by chromosome instability. So that's kind of our favorite hypothesis right now, but we have very little evidence that the chronic interferon is really actually involved in it. But so we have to do sort of the in vivo inhibition studies, but that's kind of where we're going. Yeah.
Thank you. That was super cool.
I got one random question. So there was this part of your talk where it sounded almost like P53 was not as important as everybody thinks. There were lots of correlations which were not as big as anybody would have hoped, except for the one with chromosome losses. So I was wondering, how many correlations did you actually do? Because with my statistician hat on, I was wondering, maybe there is no correlation that's actually significant. It's just you did so many that the one with chromosome losses just happened to be, it's just the one randomly a significant one. So did you correct for multiple testing, or could this also simply be a spurious correlation?
It could be.
So there's nothing going for T53.
Well, I mean, so clearly if you collapse everything together, then the degree of aneuploidy really, is significantly associated with P3 mutations. What I think is interesting is it's kind of a dogma in the field, right? That P3 mutations, aneuploidy, those are that's the tolerance mechanism. We all know that can be the whole story, but I think what is kind of interesting is that there's very clearly cancer. I mean, this endometrial cancer, for example, is a very beautiful example where you can have very high levels of aneuploidy, P3 mutation or not, doesn't seem to matter at all.
Right.
And so what is like, what is it? We don't think about that tissue specificity in p53 responses and aneuploidy very much, I think. And this would basically inspire us, at least, to look much more into how does p53 respond to similar types of stresses induced by aneuploidy in these different tissues? Is there really something fundamentally different there? And could this explain part of that? And then the other part is like, what other mechanisms for this, dealing with these stresses is out there? The losses has been also suggested by Susanna Storkova in a paper where she forced monosomies in cells. So I think it might make sense. There's some ideas about why losses and the genes that are on that are sort of ribosomal genes and protein sort of translation genes and things like this are like more sort of spread out across different chromosomes. So maybe there you would have haploinsufficiency for those genes, and that causes sort of a p53 activation. So I think also there are some interesting hypotheses about why specifically monosomies are more associated with p53 mutations. But yeah, it could be that actually you really have to look for the correlations to see the significance. Yeah.
Good, Geert. Thank you very much. That was excellent. Thank you all for joining. If you look in the chat, I just posted the schedule for the rest of the year. The next talk will be Ofer Shoshani on May 8th. And if you again look in the chat, there is an exciting conference coming up in November in Berlin. And I hope to see many of you there. With that, thank you all very much. Geert, thank you so much. And
thanks everybody for logging in. Thanks, Florian.
Bye-bye.